Time. Okay, we're being recorded. Y'all look nice. I have on zero makeup today. Just been like super busy. So. Zero. Now that I have glasses, it kind of hides it. But... Okay, get that off. And then we can go to slides. And I'm going to minimize this thing because I think it's also recording that. All right, so we're starting one of my favorite sections today, which is viruses. And if I could teach any specialty topic upper level class that I wanted to, it would be about viruses. And I haven't gotten to do that yet, but it's one of my favorite things to think about. So we know from previous time in this class, and we discussed that viruses are not living. We consider them a microbe, but they're not living. And because they're not living, that changes how they operate in many ways. But I want to start off with this question about if we think about a successful virus, what makes it successful? Is successful dangerous? Or is successful something else? And I would say to you, you all have grown up during COVID era. The COVID-19 viruses has been one of the most successful viruses in the world. Why would I say that?
It's spread to the most people.
It's spread to the most people. So for a virus, what makes it successful is the total amount of replication, which is copying itself, and spread. So it's not whether it kills a lot of people or kills a lot of animals or plants. How far did it spread, and how many total of that virus are present on the earth at a given time? So we could kind of think of that as like, if a virus had a goal, they don't. But if they did, their goal would be as many more of that same virus as possible. And COVID-19, we'll talk more about it next week, but it was especially good at spreading. It is not one of the most deadly viruses ever from its rate of people killed as a percentage, but the amount of spread was staggering. So the way that viruses spread and the way that they affect our health, we're going to learn about that during this unit. So we could talk about viruses that are famous, or viruses that are successful, or viruses that are especially deadly. And one of the more deadly viruses is one called Ebola virus. How many of y'all have heard of this ever? Did you ever watch a scary movie about a virus like that? I don't watch those movies because I know too much about this stuff and I don't want to think about that aspect. But Ebola is very lethal. This goes with Ebola info. Ebola is very lethal, but it has had limited spread. Partly because if you get infected with Ebola, you get so sick that you can't walk around and spread it to other people. And it's also obvious to the other people that you are very sick. It doesn't hide. It doesn't hide. So this part is about SARS-CoV-2, which some people call COVID as a virus. It is not very lethal. And it has had enormous spread. has had an enormous spread. So back in 2020 and 2021, as someone who kind of specializes in viruses and immune systems and microbes, you know, I was pretty obsessed with reading news about COVID-19 and the vaccines and what was going to happen and what can we do. And I remember a day when I got really cynical. I was like, oh gosh, this is never going to go away. And it was the day that there was a news report that COVID-19 had spread into wild deer populations that were running around in the woods. Like there were deer that were catching COVID and some of them would die from it and they have COVID now. And that's kind of when it sunk into me like, oh, this is never going away in my lifetime. If it has spread to the deer in the woods, and then, you know, you'd see reports like there's zoo animals dying of COVID. No, never going away. So it spreads everywhere, low percentage of death rate overall. Ebola is so deadly. It doesn't hide, and people, it ends up not being killing it. It ends up killing a higher rate of people. Hang on, I got this. I need to rephrase this. Okay, so most of the people that would have Ebola would die from it. It's a high percent lethality rate. But this situation has led to fewer deaths overall because of the limited spread. So if we compare that with COVID-19, of the people who get infected, few die because it has a low percent lethality rate. What do you think the lethality rate was or is? Let's say from years 2020 through 2022. What percent of people in the world that got COVID-19 died from it, would you guess?
1%.
It would be ballpark 1%. It's probably closer to 0.5% when you account for all ages. So if you're very elderly, it's way higher than 1%. If it's your all's age, it's way lower than 1%. If it's little kids, even lower. But probably we're going to put 0.5%. That's not 50%, that's maybe 0.5%. And then with Ebola, it's probably more like 70%. But overall, there have been millions of people dying from COVID-19 overall because it spread so much. It spread so much. And you all may have lost people in your families to COVID. There were several faculty and staff from 2020 to 2022 that lost their dads. So Chaplain Alderman, his dad died of COVID. And our previous director of undergraduate nursing, she had moved to a different institution, her dad died of COVID. And even though individually it's not that deadly, it spread so much that it caused massive numbers of deaths. And viruses don't have to be scary to cause huge public health effects. So in some ways, I don't know, how many of you all in a previous class have learned like details of COVID virus? Okay, we will talk about them in here. Like after a while, I kind of was tired of talking about it so much because people would ask me like, how's this work? I felt like I was talking about all the time. Kind of tired of it, but you do need to know the specifics. And so, what we will cover in this class is our highlighted viruses are the two that are most relevant to your nursing careers in the next 10 years, which are influenza, which this year, people as a group, their COVID immunity is such that we're getting to the point where influenza and COVID-19 are gonna be more equal in their effects than the last 2 years. If I had time for a third, which we don't, it would actually be HIV. So when I was your all's age, that was the virus that was, if you had time, you learn about HIV. Those three are the most important right now this year. Ebola is super scary. It does not really affect health in the US at this time, so. But even since I've been a faculty member at King, the first one to come through was something called Zika. You heard of that? Y'all would have been babies then. Zika virus scare, Ebola virus scare, COVID-19, which is unbelievable negative impact on the world. And there'll be another one before you all become 40 years old. That's just how it goes. So really important to learn about viruses. Okay. So there are going to be several times coming up in this class where your book teaches a concept by use of a long listy list. And the information in the list is very good, but there are things that I want you to highlight more than others. So in this list, all of this is true about viruses, but there are certain ones that I want you to focus on more than others. So the first one is to remember that they are not living. They are not living. And that's related to this fact that they are obligate intracellular parasites, which means that they're going to depend on a host cell. They have to have a host cell to build more viruses. A virus is totally pointless and it doesn't start to matter until it gets inside the host cell. We know that we can be hosts for viruses. Plants can have viruses. Algae can get infected by viruses. So can a fungus, so can a protozoa, so can a bacterium. Viruses can infect, there is no type of life that can't get infected by some kind of virus. We're gonna highlight what they're made out of. So today's lecture is gonna include this info about viral pieces, or what builds a virus. And we'll learn that list in the upcoming slides. And then this part has to do with viral life cycle. Like how do they do what they do inside a host cell? So we're gonna learn 5 key pieces of a virus. And then there are also 5 different steps in a viral life cycle. And both of those 5, I want you to memorize. So there's gonna be a memorizing list thing, but sometimes people get them backward because it's both 5 things and it's both about viruses, so be careful. Make very clear, am I talking about pieces of virus or am I talking about stages of what happens in a virus's existence? So in general, viruses are way smaller than other life forms. So we've kind of got the range of sizes of viruses are about like this. And the very biggest virus, we're going to struggle to be able to see it under a microscope. So we don't attempt to see viruses in the lab. As a group, they're just too small. So generally, they're a lot smaller than bacteria. And you really need an electron microscope to be able to see them. So as a group, viruses are really diverse, and we can talk about them in a variety of ways. So we can talk about viruses according to what shape they have. Like if we have a piece of a virus, what general shape does it have? And those 3 shapes are called helical, icosahedral, and complex. If a virus is helical, it's going to look like a stick or maybe a bent wire. So it's basically a cylinder, and it might be a straight cylinder, it might be bent into a shape. So look back at Ebola. Ebola is helical, but it's bent into it has curves. It's bendy, still helical, extended shape. This one is a plant virus called tobacco mosaic virus. It's pretty much a straight stick-looking thing, very small. The second shape is called icosahedral. So it's going to be symmetrical and have multiple sides and kind of look like a 3D geometric shape. And most viruses, you can look at it, it's like, okay, that's helical, that's icosahedral. And then if it's neither of those 2, we call it complex. So a complex virus is going to have no symmetry to it. A helix, if you cut it in half, at least the 2 sides would match. But a complex virus, neither of those other 2 shapes. And you can make a note on here, influenza and COVID-19 or SARS-CoV-2 are both icosahedral viruses. Okay, so now we get into, okay, what are the 5 different pieces that a virus can have? There are 5 different particles or pieces that can build a virus. And I do want you to memorize these. So we're going to write this list over here on this board. Okay, so the first one. Every virus has some type of nucleic acid which we consider its genome. Every virus has some type of genome. And whatever the genome is, it's going to be in the middle of the virus, like kind of deep in the core. And what the virus is, but the nucleic acid is made out of, can either be DNA or it could be RNA. It's not going to be both at the same time. It's one or the other. And so our genome is DNA. That's normal for what we think of genes are in the DNA. Some viruses have RNA genome. And for any individual virus, whatever type of nucleic acid it has, it's either going to be single-stranded or double-stranded. So they can, as a group, they have really diverse genomes. So when we divide viruses up into their big categories or their big groups, we divide them up based on what type of genome they have. So do they have a double-stranded DNA genome? That set of viruses goes into a big category together. Or the ones that have a single-stranded RNA genome, they go into a big category together. Viruses, when we name them, they don't have genus and species naming. That's for living things. So they're classified into families, and then they have strain types, but they're not really a genus species naming. And their names tend to have numbers in them, which is less easy to learn, I think. Okay, so that's piece number one. They have to have a nucleic acid. Looking back at this picture, this red line, that's the nucleic acid. So it's in the middle. And in this picture, this twisty line, that's the nucleic acid in the middle. And in this one, here it is, nucleic acid in the middle. So all viruses have that. The next thing that all viruses have is a protein coat that protects the genome. And we call that capsid protein. C-A-P-S-I-D, capsid. Every virus has at least these two things. A genome and some type of protein to go around the genome and sort of protect it. So that's called capsid. So in this particular virus, there's DNA genome on the inside, and then there are these look like, I don't know, wasp eggs or something, proteins that are repeating and forming this shell around the genome. So these are the capsid. Looking back at our previous ones, these yellow things are capsid. And on this one, the colorful shapes that's capsid. So whatever is kind of right next to the genome on the inside, we consider those capsid proteins. In HIV, here's the genome, here's capsid around it, and then there's some other stuff on the outside that has different names, but every virus has at least these two. Those are required. But for some super simple viruses, that's all they have, just those two things and nothing else. So tobacco mosaic virus is just genome and capsid. And human rhinovirus, which is common cold, is just genome and capsid. But some viruses have other structures outside of that. So these next three, not all viruses have them, but some of them do. And our third component is something called an envelope. And if a virus has an envelope, it's a lipid bilayer membrane. That actually came from the host cell. So the virus gets some host membrane as it leaves the host cell, and it wraps it around itself, and then it has on the outside this membrane. It's pretty much like an animal cell membrane. Not all viruses have that. So here's an example. HIV does have an envelope around it, which is a membrane. And inside that, it's got the capsid, and inside that, it's got the nucleic acid. So if viruses are supposed to have an envelope, they have to have it for their own life, but not every virus has an envelope. It's just a membrane, but it's called envelope. I'm gonna erase and then talk about number 4 and number 5. Number 4 is also optional, but also common for viruses, and that's something called spike proteins. Spike protein. So these are examples of spike proteins, and they stick out like a spike. And the spike proteins are super important for viruses that have them because they stick out from the surface. And the spike proteins control, they're going to control which host cells can get infected. Let's make this a witch. So the spikes kind of stick out, and they're kind of like a lock and key concept that they have to bind onto something in the host cell for the virus to infect that cell. Spikes always stick out on the surface. These viruses don't have any spikes. Not every virus has them, but a lot of viruses do have them. And then lastly, the fifth component, which is the most rare, is viral enzymes. Some viruses carry around inside them, as part of their package, some enzymes. And if they have those enzymes, those are gonna be needed early on in the infection process. This is kind of if you're an advanced virus, you might carry around enzymes as a part of your package. So HIV does carry enzymes around with it to help it infect the host cell right away. But that's kind of rare, and influenza and COVID-19 don't think they carry enzymes with them. So those are the parts. The first two, all viruses have to have. This next three, some viruses need, but not all. HIV has all of them. It's an advanced type of virus. This one, I have a doll of it in my office. I have to show you. I love my bacteriophage doll. Can I pass this around? This is a virus that only infects bacteria. I love that thing. I about had a panic attack just now because I used it. I used to have it hanging on a hook in my office. And so this is not one that could ever infect a person because its host cell is a bacterium, and that's how it looks. It's a complex shape. So it's got genome, and then it's got capsid around it, and that part's icosahedral, but then it's got all of these other parts that help it infect a bacterium. And all of these other parts are because if you're gonna infect a bacterium, there's a cell wall right here. So getting past a cell wall is not easy. And we'll learn a little bit later, like how does it get past the cell wall and how does it infect a bacterium? But that's one of my favorite things to teach about. We learn more about it in bio majors class than in here, but it's still very cool. Oh yeah, somebody made it like squat down legs. I also have a doll of an antibody and a platelet. I don't know how I got the platelet. It was just laying around in the department, and eventually I said, I'll take that platelet. It's boring compared to this. So this picture is showing, just reiterating the concept that all viruses have a genome. All viruses have some type of nucleic acid, and it can be DNA or it can be RNA, and it can be single or double stranded. So this is kind of showing you, here's some examples of viruses that have different kinds. And I want to circle one up here that is called parvovirus. Talk about that as an example. So most of the ones up here, maybe you've heard of. So we've heard of HIV. Maybe you've heard of rotavirus. You will when you get into nursing because it causes some pretty serious infections in babies and then old people. Rotavirus. And that one has RNA genome that's double-stranded. We've heard of influenza. You've probably heard of polio, more back in the day than today. Herpes, variola, that was smallpox. Parvovirus, we don't so much tend to hear about. So there is a virus that infects dogs. It's called parvo. And you're supposed to vaccinate puppies against parvo because if they get this virus, they're going to die from it. It's really sad. It's like a neurological bad death for a dog. People cannot catch that version. Like you could hold your dying puppy that's having parvo and hold him while he dies and he could lick your face and you're not going to catch parvo. Because you can't catch parvo, because that virus can't get into your cells. So there are so many viruses in the whole world and just so few of them actually infect people because you have to have the correct spikes to get into the host cell. So lots of viruses exist. Most of them don't infect people. This slide, you can write those words down, but I'm not teaching this concept right now. So make yourself a note. This is like not on the test. Sometimes I get real optimistic when I make slides. I'm like, yeah, I might have time to talk about that. Not this year. Not this slide. We might come back to it when we talk about influenza. Okay. So this picture is showing, highlighting again, what's important about spike proteins. So if a virus is actually going to infect a cell and get inside it, a lot of times it's using its spike protein that's sticking out to help the virus find a receptor on the host cell. And these two have to have a shape match. They have to have a shape match to get inside the host cell. And if the shapes don't match, you could have 1 million viruses out here right next to this host cell and they don't have a way in. That shape match is required. So if the virus has a spike and the spike doesn't match with a host receptor, it doesn't matter how many viruses are out here, they don't have a way in. So I don't know why I would do this, I could purify 1 billion bacteriophage or 1 billion parvovirus and eat it or snort it or put it in my vein, and I'm not going to catch that because it has no way to get into my cells. So that's what controls host range. I could eat tobacco mosaic virus as nutrition. Bizarre, but I could use it for calories, eat it all day long. I'm not going to get infected with it. So the spike is really key for infection. Okay, so those are the pieces. Those are the parts of viruses that can exist. And once we know what the parts are, then we can start learning how do those parts help a virus infect, and what is a virus actually doing to the host cell. And we call this viral life cycle, which is kind of messed up because they're not alive. But viral life cycle. So it's kind of like, what are the steps in the progress of a virus? And these are their names. So we've got attachment. We've got attachment stage. Second is called penetration stage. The third step, most books just call it synthesis. This book, OpenStax, calls it biosynthesis. Either is okay with me, but most books call it synthesis, and I tend to call it synthesis because, all the other 4 or 5 books I've taught out of before OpenStax called it that. That's how I learned it. This stage has 2 different names depending on the book. It can be called maturation or assembly. And I learned it as assembly. You can write down either one, but I'm going to call it assembly. And then the last stage is lysis. Or release. Lysis or release. And what we're going to do on this slide is kind of underneath these boxes write a short description of actually what is happening during that stage. And I need to fast forward a bit and see if I've got. Okay. That's what I want to do. So that is what I want to do. So step 1, attachment. In attachment, the virus must bind to the host cell and usually it's using its spikes to bind. And in a human system, that's pretty much always the case. So the virus has to attach onto the host cell. It's not just hitting the host cell. It's not touching the host cell, but it's a specific shape match attachment like this. They got to match each other in their shapes. So after that happens, the next step, the virus is hanging out there and it's not just hitting the cell, but it's attached to the cell. The next step is the virus or at least the genome. This is different depending on what type of virus we're talking about. Either the whole virus or maybe just the genome has to get into the host cytoplasm. And then once that happens, you can assume that the virus is then going to take over the host cell. That's penetration step. This cartoon up here is showing how does bacteriophage do this stuff to a bacterium. So they're using that as an example. So first, the bacteriophage has to attach to the host cell with these little tail fibers. Just attach. And then, because there is a bacterial cell wall here, it's hard for the whole virus to get into the cell, so it just sends the genome in. And so it looks like it's pooping it out. All of this stuff is to help the genome get from the head. Like, is it smiling? No, it's emotionless because it's a virus. The genome has to get from the head down through this section, and it actually makes a hole in the wall and it shoves that genome in there. And then the genome takes over the cell. We'll look at how influenza and SARS-CoV-2 do that next week. So the genome is in, the genome is going to take over. And that's the synthesis stage. So in synthesis, the viral genome takes over the host cell and it makes the host cell build viral pieces. So the host cell then becomes a virus factory. That's really sad for the host cell. So instead of the host cell doing what it ought to be doing that day, it's forced to build genomes and all the different other pieces of viruses. And so in this step, the host cell is going to use energy. Like it spends pretty much all its ATP on virus work. So it's going to use its energy and it's going to use its resources. So use my own amino acids and my own membrane lipids and my own nucleotides to build virus stuff instead of doing the work I ought to be doing. So this is bad news for the host cell. And then so once all the pieces have been built by the host cell, during the assembly stage or the maturation, the viral pieces self-assemble so that they can form new viruses. That's also really cool. Like, there's all the pieces in there and they automatically form themselves into new viruses. So that's kind of, you can see that here. Over here we've got just pieces. And then by this point, the DNA has gotten into the capsid and the viruses are now pretty much finished. That's the assembly or the maturation. And then in the last step, the virus is going to leave the cell. Virus is going to leave the host cell. It should say viruses leave. Let's change that. The viruses leave the host cell. And in this step, there are two different options, and we'll talk about that on a different slide, because it's easier to show on that different slide. The viruses are finished, viruses leave, and then circle this where it says viruses. And those viruses can then go and infect more new cells after that. Those 5 stages are done. Key slide here. So the way, looking ahead to the test, which is not for a while, but this type of question is like, list the 5 stages of the viral life cycle, part A, 5 points or 6 points or something like that. And then the other questions that are coming after is like stuff about what's happening. So like, which of these stages, blah, blah, blah. That type of stuff. The one about the viral parts, I usually just have you write that down in a list. Like you need to know the parts because the parts relate to all this other stuff. So the stages, you tend to get a big question about stages. The parts, you need to have those memorized also, but it's because they affect this other stuff. Okay, so now we're going to talk about the viruses leaving the cell. And I'm going to turn the lights all the way off for this. This is one of my favorite biology pictures. You probably don't have this in OpenStax, right? It's a real shame. This is an old picture. This is an electron micrograph that's showing E. coli. So if you got this picture, you can draw around the rod-shaped oval here. This is an E. coli cell. And out here, we've got bacteriophages. Like they're just totally lined up outside this cell trying to infect it. What they don't know, because they're dumb and they don't have a brain, is that this cell is already infected. And in fact, this cell has already built a bunch of viruses which are now bursting out of the dead E. coli. Here they come, they're spilling out into the environment and they're going to go infect more E. coli. So is this terrifying? Is this awesome? It like depends on your outlook. I kind of, intellectually love viruses, and this is both scary and it's also cool. And fun fact, bacteriophage, if one of them lands on an E. coli cell, it can kill it in 14 minutes or something. It's crazy. 14 minutes from attachment to release. So this type of release is called lysis. That's the first style, lysis. Lysis is when the host cell bursts open so that the viruses can get out. It's lysis. So for bacteria that have a cell wall, there is no graceful leaving of the virus. The virus has to kill you to get out. That's one way to get out of the cell. It's not the only way. So in viruses that infect humans, there's another way that's maybe more common. And this will be our last slide for this day. And the second way, you've already heard of this, it's called exocytosis. So probably you learned that in high school biology, where a cell releases something outside of itself in a little blob of membrane. And endocytosis is the opposite. So a virus that infects a human a lot of times will trick the human cell into endocytosing it. So influenza virus, we would have the spikes bind, And then the penetration step is endocytosis. So the host cell, like, oh, yes, okay, I've got something out here I need to bring into the cell. So it comes into the cell with membrane wrapped around it. And then it uncoats itself and the virus starts to take over the cell. And it makes the, here's the genome taking over. It makes the host cell do the assembly. And then when the virus gets ready to leave, it buds off of the cell and it takes membrane with it. So here's the virus. It actually takes some membrane from the host cell with it as it leaves. And so you can call that exocytosis or budding. So some people would refer to that, the virus is budding off the host cell. This might not kill the host cell. It might kill the host cell, but it might not. It just kind of depends, like how tired is that cell and how many viruses tried to leave it all at once. It might still live, but it might be so worn out that it dies 10 minutes later. Just depends. So that's the other way a virus could leave the host cell. This is Wednesday. On Friday, we do not have class. Not this class. You might have other classes, but not this class. So the next time I see you guys will be Monday. So have a good rest of your week and a good weekend, and we will finish whatever's left in this slide set on Monday, as well as go to the new stuff. Not too much.